Today we're going to be talking about the process of mitosis. And one thing I want to clear up before we go any further, there's a couple different ways you can identify mitosis. Technically, mitosis is the division of the cell's nucleus. And then it's followed by cytokinesis. But the way we're going to be talking about it today and the way that it's normally presented, when we talk about mitosis, we're just going to throw cytokinesis in there and assume that it's the division of the cells. So truly mitosis is just the division of the cell's nucleus, and then it's followed by another process that separates those two cells. But like I said, in most textbooks, and the way we're going to be talking about it today, we're talking about the asexual reproduction of a cell. One cell becoming two cells. So before any of that can happen, we have to divide the nucleus, and before we can divide the nucleus, we have to replicate everything inside of it, because remember, the nucleus contains DNA, which are instructions, and those instructions must be replicated before we divide the cell in half. If we divide the cell in half without dividing the nucleus and replicating everything inside of it, then we wind up with two cells. They each have half the instructions that they need, and the cells will die. So they have to have a complete set of instructions. So we're going to replicate that DNA, then divide the nucleus, then worry about the rest of the cell. So when that nucleus is being divided and when that DNA is being replicated, that's referred to as the M phase or the mitotic phase. So we actually take care of DNA replication before we go into any part of cell division. That's part of the cell cycle. It's the S phase. We're going to get all of our DNA replicated, basically get all of our ducks in a row, get everything set and ready to go. Then we can enter this where we actually start moving things around. So mitosis is going to be used in all of our cells with the exception of our sex cells. So our sex cells undergo a different process, which is referred to as meiosis, to become sex cells. But every other cell in our body undergoes mitosis. It's how we get more cells. You are a much larger organism now than when you were 3 years old. That's not because you now have adult-sized cells. It's because you are made up of more cells. And the reason that you are made up of more cells is because those cells have undergone mitosis. So you have more bone cells and more skin cells wrapped around them and more muscle cells and everything else. And having more cells makes more of you. So once we have the DNA replicated during that S phase, everything's good to go. We're going to be prepared to go through the rest of our process. So we're going to look at this. There are four main phases of mitosis, or four main stages of mitosis, and we're going to look at each one of those individually. So if we're talking about humans, what we're going to really see is we're going to have a cell that has 46 chromosomes. It will temporarily have 92 chromosomes, and then we will divide that in half, and you'll have two cells, each having 46 chromosomes. You never really see the 92 chromosomes in textbooks and things like that. They're not going to call it that. They're just going to say that it's everything's been replicated. So usually you don't see those numbers to illustrate it, but that's what's happening here. You have to go from two sets of chromosomes to doubling that, right? So it's, technically four sets. Then we separate them, and we go back to having each cell having two sets of chromosomes. So we're going to look at all the different phases that we have here. So before we can start anything, the cell has to replicate its DNA. All right, so we're going to look at a little diagram as we go through these phases here. This is a simple diagram representing the four stages of mitosis. So the first phase we have is referred to as prophase. Okay? Prophase is right over here on the left. The chromatin fibers are going to become more tightly coiled and folded into compact chromosomes. So this is where you see these blue lines here. These are our chromosomes. Normally, inside the nucleus, you only have chromatin. Chromatin is a thin, wispy material. It's a lot like thread. I always say that chromatin is like thread. If chromatin is like thread, then chromosomes would be like spools of thread. It's where we condense all of our genetic information, makes them really easy to sort. A lot of people always ask, well, if it's so easy to sort, why don't we just always have chromosomes? Well, because chromosomes, because they're so tightly packed, they're very hard to access. So when we need to transcribe a gene, it's going to be really tough to find it when all of your genetic material is packed together. So we leave all that material unpacked in the form of chromatin. That way it's really easy to access and really easy to transcribe. So other things that are going to be happening in addition to this chromatin condensing into chromosomes. you're going to start to see the disappearance of the nuclear membrane. So the membrane is what separates the nucleus from the cytoplasm. That's going to start to disappear. These sister chromatids are going to become visible. These yellow lines, those are spindle fibers. Those spindle fibers are going to start to form, and these molecules that hold them here, these little centrosomes or sometimes centrioles, they are going to hold the spindle fibers. Okay, so all of that's happening during prophase. As we phase into metaphase, again, that nuclear membrane is going to start to disappear, and then we go into metaphase. Metaphase is probably the most easily recognizable phase if you're looking at a diagram. Okay, when we look at a cell, we think of a cell as kind of like a globe, right? And a globe has a north and a south pole and an equator. Okay, a cell is similar in that we refer to the middle of the cell as the equator. You'll notice here that the chromosomes have now lined up along the equator of the cell. Okay, the centrioles have moved to opposite poles of the cell, and our spindle fibers are now reaching out and grabbing those chromosomes in the middle. So like I said, when you look at a diagram, you can almost always spot metaphase because you're going to have that distinct line of chromosomes along the middle of your cell. The next phase that we move into is referred to as anaphase. In anaphase, you've noticed the cell's gotten a little more elongated, right? So it's not a perfect circle. It's an elongated shape at this point. The centrioles, they start pulling the sister chromatids to their respective poles. So here, what you have in this image is you have chromosomes that are connected like this. That's giving them the distinct duplicated chromosome, or the X shape. What we're going to do when we start pulling them away, though, is we're going to connect right there at that centromere, okay, and we're going to start pulling them towards opposite poles. So now instead of X's, they look like V's, right, and they're moving in opposite directions. So all of that's going to be happening during anaphase. They're going to continue to pull these chromosomes, or now they're really chromatids, back to opposite ends until they reach the centrioles. Once that is completed, we enter the last phase of mitosis, which is referred to as telophase. We're going to continue to elongate the cell, so the cell is going to get a little bit longer. We're starting to see the formation of nuclei at either side of these two new cells that are starting to form. We're going to start putting a nuclear envelope. Essentially, what's going to happen during telophase runs in contrast to what you have happening during prophase. So in prophase, you see the disappearance of the nuclear membrane. You see these chromosomes start to condense. In telophase, in contrast to that, you see the reappearance of the nuclear membrane, and these chromosomes are going to start to uncondense back into chromatin. So you're going to see the loosening or uncoiling of that material. You're going to see the reappearance of the nucleolus, which also disappeared during prophase. You notice the mitotic spindles, or those spindle fibers, they are now gone. And also you can start to see a little bit of a pinch starting to happen. This is called our cleavage furrow, and we're basically going to take that cell, if it's an animal cell, and if it's like this, and we're essentially going to just squeeze the cell like this and pinch it into two cells. And because the cell doesn't have a cell wall, that's really easy to do. If it were a plant cell, if we're looking at a plant cell, because of the presence of a cell wall, we're not going to be able to pinch the cell apart because the cell wall is too rigid. So what we're going to do instead is we're going to form a cell plate right down the middle and push that cell in that direction and push that new cell in that direction, and wind up with two new cells. So we're pinching those cells in the middle of right there in the middle during telophase, and once they have pinched, then we're ready to enter. It's no longer considered a phase of mitosis, but it's how we wind up with two new cells, and that is called cytokinesis. And that is where we actually separate the cytoplasm and we have two new cells. So the end result here is we have two identical cells. We took one cell that had 46 chromosomes, okay? And we replicated everything inside that cell, and so we temporarily had 92 chromosomes. And then we simply divide that cell, and we now have two cells that have 46 chromosomes. Or another way we represent that when we're talking about sets of chromosomes, we start with a cell that is diploid, so that's 2N, right? 2 sets, and we divide a cell and we get 2 cells that each have 2N. So my question that I get from students a lot is, well, you can't divide 2 by 2 and get 2. Right, because temporarily, you'll never see it in a textbook, or temporarily, this is actually 4N, because we replicate everything inside there, and then we divide. So these 2 new cells are identical to this one. They are made up of, this one just expanded and divided. Okay, and so this is how we get new cells. This is how we grow. When you get a cut on your arm and you need to replace the skin cells that were there, mitosis is the process that's going to do it. So growth and repair is what mitosis is all about. Organisms are undergoing it all the time in order to repair those damaged parts and to get larger. At a certain point, most organisms reach a maximum size, but you can't stop mitosis there because you're still going to have lots of repair from injury and just wear and tear that you're going to need to do. You're going to need to replace those cells throughout your body, and mitosis is the process that's going to do it. I'll see you next time.